Today I will be reviewing the process of adaptive immunity, which is when the body develops immunity to a specific antigen only when it has been exposed to that antigen before. Specifically, I will be going over the antibodies and B cells within this process. I will begin with the antigen-antibody complex. Antibodies do not bind to the entire antigen, but specific areas called epitopes. A complete antigen has at least two epitopes, one for each of the binding sites on an antibody molecule. Sometimes antigens are not complete, which result in haptens, or partial antigens. Haptens can join with a carrier molecule, making it a complete antigen, so an antibody can now bind to it. Moving forward, I will review the sensitization and activation of B cells and the role they play in adaptive immunity. We first begin with the sensitization phase when the inactive B cell is exposed to antigens. Here we have an inactive B cell with antibodies and class II MHC proteins attached. When antigens come along, they bind to the antibody molecules. The class II MHC protein then presents an antigenic fragment on the plasma membrane. This tells the other cells that there are pathogens, toxins, or foreign proteins in the body. The B cell is now sensitized. Next, the activation phase occurs, where the B cell meets up with a helper T cell. The class II MHC protein binds with the T cell receptor, making the T cell receptor recognize the antigen and begin secreting cytokines that promote B cell activation. After activation of the B cell, cytokines also stimulate B cell division, producing plasma cells and memory B cells. Plasma cells synthesize and secrete large quantities of antibodies, while memory B cells are made to remain in reserve to deal with injuries or infections that involve the same antigens. If they did see the same antigen, they would divide and differentiate into plasma cells, which would then secrete antibodies. Now that we know how antibodies are made, we can now learn the 7 ways in which they can cause elimination of antigens. First, neutralization, which means that antibodies bind to specific parts of the body that viruses and toxins want to bind to, making it impossible for them to do so. Next, precipitation, which is the formation of insoluble immune complexes when soluble antigens such as proteins react with soluble antibodies. Another elimination of antigens includes the complement system, where after an antibody binds to an antigen, portions of the antibody change shape so complement proteins can bind, activating the complement system to destroy the antigen. Also, phagocytosis, which is when the antigens are covered with antibodies attracting eosinophils, neutrophils, and macrophages, which then destroy the antigen. The 5th way is opsonization, which is a coating of antibodies and complement proteins, which make it easier for phagocytes to hang on to their prey. Antibodies also stimulate inflammation by stimulating basophils and mast cells. Lastly, antibodies prevent adhesion by dissolving in saliva and mucus, adding an additional layer of defense. We have reached the end of reviewing adaptive immunity, explaining the roles of B cells and antibodies. Thanks for watching.